Have you ever wondered how two parents can produce multiple offspring that look completely different from each other? Or how it's determined whether you're biologically male or female? All eukaryotes reproduce through a method called meiosis. In this short presentation, we'll explain what meiosis actually is and why it's so important. Then we'll take you through the process of meiosis step by step. There are two types of cell replication. Mitosis is a process where one single cell divides and creates two identical copies of itself. Almost every cell in the body is created through this process. Meiosis, on the other hand, is a unique type of cell division, resulting in 4 genetically varied cells with half the number of chromosomes. While mitosis only has one division, meiosis has 2. Within each of these divisions, there are 4 unique phases: prophase, metaphase, anaphase, and telophase. So why is meiosis so important? Meiosis is essential in increasing the genetic variation of a population. It is through meiosis that humans are genetically varied from one another and not mere clones of their parents. Another important aspect of meiosis is the determination of sex. As we will learn soon, during the process of meiosis, the baby's sex is randomly determined. Humans have 46 chromosomes, which can be a lot to keep up with. To keep things simple, we'll be showing the process of meiosis with only 8 chromosomes. This is the same number of chromosomes that fruit flies and some species of flowers have. For our presentation, we'll demonstrate meiosis through a male sex cell and mark one chromosome as the X and one as the Y. During the final stages of interphase, the cell duplicates its DNA and prepares itself for cell division. These steps are very similar to what would happen to a cell during mitosis. The first step of meiosis begins with prophase 1. During prophase 1, the genetic information begins to condense and form distinct chromosomes. Next, each chromosome aligns itself with another chromosome, called its homologous chromosome. Homologous chromosomes are similar in length, centromere position, and staining pattern, and contain different alleles for similar traits. For example, one chromosome in a homologous pair might code for blue eyes, while the other codes for green. Then, the homologs undergo a complex exchange of genetic information called crossing over. This shuffles up the DNA to create unique combinations of alleles, which leads to greater genetic diversity. The next step is metaphase 1. During metaphase 1, pairs of homologous chromosomes are arranged along an imaginary line at the center of the cell called the metaphase plate. When the chromosomes line up, their orientation is completely random. This means there are literally millions of possible combinations of alleles the final daughter cell might end up with, which again contributes to the vast genetic variation that occurs during meiosis. In male gametes, it is during this phase that the male sex chromosomes are randomly assorted into their daughter cells. Half will end up with an X chromosome and the other half will end up with a Y. When the male and female gametes fuse, the pairs that contain 2 X chromosomes will develop into a biological female, and those with the XY chromosome will develop into a biological male. During anaphase 1, the pairs of homologous chromosomes are pulled away from each other towards opposite ends of the cell, and finally during telophase 1, the chromosomes arrive at the ends of the cell and cytokinesis begins, separating the cell into 2 haploid daughter cells. After telophase is complete, the daughter cells begin the next stage of meiosis, meiosis 2. Unlike meiosis 1, meiosis 2 does not begin with the cell's DNA replicating. In prophase 2, again, the nuclear membrane decomposes and sister chromatids condense and become more visible. Unlike prophase 1, no crossing over occurs here. In metaphase 2, chromosomes will then arrange themselves along the metaphase plate. In anaphase 2, the centromere splits and sister chromatids are pulled away by the spindle fibers and move towards opposite poles of each cell. After the sister chromatids separate, you're left with 2 groups of individual sister chromatids. The final step of meiosis is telophase 2, when nuclear membranes begin to form around each group of chromosomes. Then, cytokinesis occurs, dividing the cell once again. The final result is 4 daughter cells called gametes.